Okay, in the previous video, we looked at transcription. That was the process where we take a functional unit of DNA called a gene and we transcribe from it an mRNA sequence. At the very end there, we were looking at how this mRNA sequence is going to be changed slightly. So it's not exactly the same as the original DNA sequence. It's going to be spliced. And so that splicing process is where we're actually going to cut out some of those introns and splice together what are called exons. So the mRNA may be a little bit shorter than the original DNA sequence because of splicing. The other things that we talked about have to happen is it has to be capped, meaning it has to have a modified G or guanine, which helps it to leave the nucleus and find the ribosome. And then the very last thing we were looking at is it has to have a poly-A tail added, which also aids its stability outside of the nucleus. Okay, so I kind of feel like that got rushed there at the very end, so I kind of wanted to hit back on those last stages that happen to the mRNA after it is transcribed. Once you have the mRNA completed, it's capped, tailed, and spliced, the mRNA is going to be able to leave the nucleus, or for prokaryotes, there never was a nucleus, so you just now have this mRNA. And then the mRNA is going to be translated. So this is what we're going to be looking at in this video, is this process of translation. Translation is where we're actually going to be moving from a nucleotide sequence to an amino acid sequence. And the amino acid sequence then will be the protein. And this is going to really be what is functional or useful for the cell. So translation is going to use this genetic code. The genetic code tells us that each group of 3 nucleotides could also be called a codon or a triplet, is going to correspond to 1 amino acid. One really interesting thing about the genetic code is that we find that it is nearly universal. There's very few exceptions to this genetic code among all the species of animals, plants, fungi, bacteria, all of the different types of creatures on this planet, all the way down to bacteria, are going to use basically this same genetic code. And I put here nearly universal. There's always going to be a couple exceptions, but it's amazing how universal this exact same genetic code is. So here's the thing about the genetic code. We have 4 nucleotides, right? We've talked about these before, A, G, T, and C. Well, as we move to RNA, the T becomes a U. So that's one little difference between DNA and RNA, is that you have a U in RNA instead of the T, and that U corresponds to uracil. It just replaces the thymine. Okay, but there's 4 nucleotides, and then there are 20 amino acids. So way back when we started looking at biomolecules, we saw that there were 20 amino acids that exist in our world. What this means is that the genetic code is going to have to be degenerate. The word degenerate here means that there's not just one codon for every single amino acid. Because if you did the math on this, with 4 nucleotides, looking at, well, how many possibilities can you arrange these into? And those possibilities would be like UUU, UUC, UUA, UUG, and so on. You know, we have 4 of them, and we could put how many possible triplets can we make from these 4 letters? We could do the math on that, which would be 4 to the power of 3, and the answer is 64. So if we add these up here in this picture, there are 4, 8, 12, 16. I could keep going all the way to 64. There's 64 possible triplets, but there's only 20 amino acids. That means that there's not going to be just one codon per amino acid. There's actually going to be several codons or several triplets that are going to correspond to the same amino acid. And if you look carefully at this genetic code over here, you can see that. UUU, UUC both encode the amino acid phenylalanine. UUA, UUG both encode, and I could keep going, CUU, CUC, CUA, and CUG, if I'm reading that correctly, all encode the amino acid leucine. So there's not just one code per amino acid, but there can be multiple. And if you really look carefully here, you'll see that most frequently it's the first two nucleotides that are going to determine the amino acid. But that's not a rule to run too far with, because there are going to be some exceptions to that. Main idea, though, is that the genetic code is degenerate. There's more than one codon for each amino acid because there's more possible triplets than there are amino acids. Okay, so when we're talking about translation, we're starting with an mRNA and we're moving to an amino acid sequence. So this is after the mRNA has been fully made, capped, tailed, and spliced. And so here's our mRNA. It's got a cap, it's got a tail, and then we assume that it has been spliced. It's now going to be able to bind with a ribosome. So it has to have a ribosomal binding site, which is going to be at its 5' end. And this 5' end ribosomal binding site is sometimes also known as the 5' untranslated region, or more affectionately, the 5' UTR. This 5' region is going to be what the ribosome is able to kind of dock with to start this process. Then, what actually gets translated begins when you get to what's called a start codon. So the start codon, if I look back at the previous picture of the genetic code, we can see in here the start sequence, which is going to be the triplet AUG. And so it's not until you have a codon in the RNA that says AUG that you can begin this process of translation. So everything else that existed before you got to the AUG is considered the untranslated region, because it's just the ribosomal binding site. This is where we're going to kind of load the RNA onto the ribosome, and then the ribosome will look for the AUG and begin its process of translation. So that's the start codon. And the start codon is going to give us what's called the reading frame. And then the RNA will be read from the 5' end toward the 3' end. Okay, and so in this process we'll talk about tRNAs, and we will definitely be talking about the ribosome. First, so let's look at what we mean by that reading frame. So in this picture, we're seeing a couple examples of some mRNA sequence. Here's the upstream untranslated region of the RNA. This is the ribosomal binding site, and we can see it down here as well for our second example. And the idea is that the ribosome will not start translating until it gets to the start codon, which is an AUG. So the ribosome is kind of stranding. It's going A, nope, G, U, nope, 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 AUG. Let me begin. So the AUG, as soon as it finds that determines the reading frame, because that determines the first codon, the first series of 3. Now, once you determine the reading frame, you can then see each of the codons. So these are going to be the next codons. This, though, is why if you have a mutation, for example, in this bottom example, we have a mutation where we have entered an extra nucleotide that shouldn't be there. Oh, look, there's this U that shouldn't be there. It wasn't up here. Everything else about the sequence is the same, though. AUG, and then CAG, so that's going to be CAG, and then another CAG, CAG. So this is all the same, except for we've got this little bonus U in here. This, though, has altered the reading frame. The addition of this weird little nucleotide is going to completely change the sequence. So this is going to be important if we eventually get to talking about mutations. By just adding one little nucleotide, we have completely changed every single codon and thus every single amino acid that would be translated. Because our first, with this little U, our first codon after the start codon is UCA instead of CAG. And if you look, there's the CAG, it's still here, but we have altered the reading frame. This is an addition of a nucleotide, shifts the reading frame, and completely changes the protein. Okay, the next thing I want to do is kind of review what we know about amino acids. So we've talked about how the mRNA is going to have these codons, and according to the genetic code, each codon corresponds with a different amino acid. So here's a codon, here's the amino acid. Here's a codon, here's the amino acid. And we could verify each of these in that genetic code chart, which is kind of fun to do, but I'm not going to do right now. Now, what we need to review is, well, first let's review what an amino acid looks like. So for example, each of these amino acids is going to have the same basic amino acid structure, which means it has a carbon in the middle, an amino at one end, and an acid at the other end. So the amino is NH3. The acid is carboxylic acid. This basic structure is going to be the same for every amino acid. But what makes the 20 amino acids unique from one another is what's called the R group, or the variable region. This R group is going to be different for each of the 20 amino acids. And we could look each of them up. But when we're talking about building an amino acid sequence, we need to understand how we're going to connect amino acids in a series. Because what this is showing us is that we have methionine amino acid connected to the serine amino acid, connecting to the glutamine amino acid, connected to another serine, and so on. How do we connect amino acids to one another? Down here, this is going to show us how we connect them. So if this is one amino acid, and this is a different amino acid, and we can kind of look here, yep, here's the amide, here's the acid, this is the carbon in the middle. Here we just have an H, so this is representing glycine, but this could have just been anything. And same over here, this is actually another glycine because it's got an H, but it could have been anything if it were a different amino acid. Here we're going to look at how do you join these two consecutive amino acids together. And the answer is it's a peptide bond, which is technically a dehydration reaction. And if this sounds familiar, it should. I'm just trying to kind of review this for you for a hot second to get your head back in the game of how we are connecting these amino acids. When we remove this essentially water with a dehydration reaction, this carbon directly bonds to this nitrogen, and these guys are removed. So here we have the carbon connected to the nitrogen, and this is called a peptide bond. And so what's going to happen is we're going to use the genetic code to line up each of these amino acids, and when they're lined up close to each other, we'll be able to form this peptide bond, sealing or connecting one amino acid to the next. So the mRNA is going to provide the genetic code that tells us which amino acid to bring in. But how are we going to bring in those amino acids? That's what we're going to look at next. And this requires the existence of what's called tRNA. tRNA is a piece of RNA which has what's called the anticodon. And then tRNA is going to be specifically bound to the amino acid that corresponds to the codon of the genetic code. So tRNA just stands for transfer RNA or translator RNA. It's doing this process of translation. And the tRNA will always have a sequence of nucleotides, which is called the anticodon. And if we looked back on the genetic chart, we would find that lysine is going to correspond to the A codon. So what is complementary to an A codon? Yes, U. So this is called the anticodon because it's complementary to the codon. And then the anticodon, or the tRNA, is going to be loaded with the appropriate amino acid. And this, which is the appropriate amino acid? Well, that is what we can read off of the genetic code, but what we determined experimentally by kind of looking at how this actually happens. But it makes sense that we would have a tRNA that has an anticodon that corresponds with the amino acid that corresponds to the codon in the genetic code chart. So let's try to see this all together in a picture. Okay, first, remember, we are in the cytosol. Definitely for prokaryotes, because that's all they have, but even in eukaryotes, we're in the cytosol. That's where the ribosome is, and that's where the mRNA that we spent so much time in the last video making needs to be. So we have this mRNA, and the first thing it's going to do is it's going to bind with the ribosome. Down here in the picture, we can kind of see this. Here's our ribosome. Here's our mRNA. Look, they're kind of bound, and we're going to see this in a little bit more detail in a second. The mRNA has this sequence of nucleotides. What's going to happen is there's going to be various tRNAs floating around the cytosol. And each of these tRNAs is going to be charged with the appropriate amino acid depending on the tRNA. So here comes the tRNA that's loaded with lysine. It has that anticodon we were looking at, UUU. It's going to bind to the sequence of the codon, AAA, and basically bring this lysine into frame so that it can be bound to the appropriate amino acid in the chain of amino acids. This is going to be our growing peptide chain. So this is bringing in the appropriate amino acid according to the sequence of mRNA nucleotides. This image kind of gives us a nice summary of what we've talked about so far. So first, this is our mRNA. It includes this 5' untranslated region, which is going to dock with the ribosome. The ribosome's then going to look for the start codon. As soon as it finds the AUG, that's the first thing that the ribosome will then allow a tRNA to bind to. Down here, we're seeing the various tRNA. So here's our first tRNA. It's got the anticodon UAC, and it's charged with the appropriate amino acid, in this case, methionine. UAC lines up because it's able to hydrogen bond with each of these nucleotides in the mRNA sequence. So the tRNA kind of comes in and correctly binds to the complementary bases. So the AUG corresponds to a UAC, and it's charged with the methionine. So that brings the methionine kind of into frame. So it's going to hang out there, and then the next tRNA is going to come in with its anticodon that's going to correspond with the mRNA, and it's charged with its amino acid according to the genetic code chart. So it brings in a serine. These two guys are lined up so close to each other that we can make a peptide bond. The next tRNA comes in, and it's also going to be corresponding to the codon. It's got its anticodon, and it's charged with the appropriate amino acid. So that brings it into the frame so it can be added next. So the mRNA sequence will determine the tRNAs that can bind, and the tRNAs will be charged with the appropriate amino acid based on that amino acid, or based on that genetic code chart. And this is going to bring in the appropriate amino acids so that our mRNA sequence will give us an appropriate amino acid sequence. And you and I can look at an mRNA sequence and we can predict what the amino acid sequence would be, because we can read each codon just as the ribosome would and use the tRNA to make those amino acid sequences. Okay, so this is kind of the big picture of where we're going. Let's try to understand what these tRNAs are in a little bit more detail. So tRNAs are an RNA sequence. There's actually a gene for the tRNA sequence. It's a non-structural gene, meaning that first we make this tRNA from our DNA sequence, and we have this sequence of RNA, which is the tRNA. This tRNA sequence, then it ends up folding because it's got various hydrogen bonds based on its sequence. It ends up folding into this shape, and I do think that this shape looks a little bit like the letter T. I often wonder if that's how it got its name, tRNA, but the T does stand for transfer or translation. There are going to be various tRNAs, and they're each named according to the amino acid it carries. We can predict, though, if we look at a tRNA, we can predict based on what the anticodon is, we can predict what amino acid it's going to be charged with. And so there's going to be a unique tRNA for each amino acid. But if you remember, we said the genetic code is degenerate, meaning there's more codons possible than there are amino acids. So there's actually, for this particular amino acid serine, there's actually 6 different tRNAs because there's 6 different codons that correspond with serine. And that's generally the case for all of them. Maybe not 6, but generally 4 to 6 tRNAs for each. And you could figure that out based on looking at that genetic code to see how many codons there are for a particular amino acid. Here we're seeing, this is more of the 3D structure, so more of the real shape of this molecule. But it's always going to have this anticodon, which is going to correspond with the amino acid that the tRNA gets charged with. So this is a tRNA. It is an RNA sequence, and it's very important that it has this anticodon as part of its sequence. We have to charge the tRNA with the appropriate amino acid sequence. And so to do that, there's actually an enzyme that does this process of attaching the amino acid to the tRNA. The enzyme that does this, it's kind of got a good name. It's an enzyme, so it ends with ACE. And then it is an aminoacyl, that kind of stands for amino acid, tRNA, and it's making it, so it's synthesizing it. So it's called an aminoacyl tRNA synthetase. And there's actually different aminoacyl tRNA synthetases for each amino acid. They're named according to the amino acid. So for example, there would be an alanine for alanine, the amino acid alanine, alanyl tRNA synthetase, which would attach alanine to the appropriate tRNA. And there'll be a different tRNA synthetase for every single amino acid that exists. These enzymes have to be really accurate. They're going to depend on that anticodon of the tRNA to figure out what synthetase enzyme it can bind to, and then that synthetase enzyme will bring in the appropriate amino acid for that anticodon. Let's look at that picture in a picture. So here's a picture. Here's that great big brown blob is our aminoacyl tRNA synthetase. So this is the enzyme that does this job. Enzymes have to have loading sites for the things that it's going to help react. So this enzyme has got a loading site for the amino acid that it's able to bind to. It's also got a loading site for ATP because attaching the amino acid to the tRNA is going to be an endergonic reaction, so we need some energy. So we're going to load an ATP to provide this energy. So here we're seeing our amino acid is loaded, and then here we're seeing the ATP is loaded. And then this big old enzyme, the tRNA synthetase, has got a binding site that has a codon that's going to correspond to the anticodon to the appropriate tRNA. And here you can imagine that not every tRNA will bind to every synthetase. So this is how the synthetase enzyme can be specific, is based on this little binding site it has for the tRNA. So only the tRNA that has the correct anticodon is going to be able to bind to this enzyme, the tRNA synthetase. But what the enzyme does is it kind of lines up the tRNA molecule with the appropriate amino acid. And we know that it's the appropriate tRNA because it has to be able to match with the codon, or the series of 3 triplicate nucleotides that correspond to this amino acid. And it uses ATP. You can kind of see the proximity of these guys and for the reaction, but that ATP is used and MP is released. That means it's just mono. It's released both of its phosphates, or 2 of the phosphates. And then we have, as a result, we have attached the amino acid to the appropriate tRNA. The enzyme then can be reused and new things can bind to it, and it can begin this process all over again. So what the synthetase enzyme is doing is it's kind of squishing the tRNA together with the appropriate amino acid. But now we can call this a charged tRNA. It is now ready to go and interact with the ribosome to bring this amino acid into frame. Okay, so let's talk about where we've been so far. In this video, we've been talking about this process of translation, and we've said that for translation, we need to have a tRNA charged with the appropriate amino acid. We've now talked about how we get that tRNA charged with the appropriate amino acid. The other thing that we need to talk about is the ribosomes. And so that's going to be the subject of the next video. We'll start to look at the ribosomes that actually bring everything together so that we can make the protein. So in this video, we talked about the ribosomal binding site at the 5' end of the mRNA. This is also called the 5' untranslated region, which we'll continue to see when we actually bring this mRNA to the ribosome. Then we talked about the reading frame. mRNA is always read from 5' to 3'. We kind of see that 5' to 3' designation both for DNA synthesis and transcription, and we're seeing it again for translation. And then we've talked about the tRNA. We just need to look at the ribosome now. And that reminds me, before I forget, I'm going to give you the password this week. Ribosome with no S is our password. It should be the right number of characters, more than 4, less than 10, all things that I've learned. RIBOSOME is going to be our password. And we'll look at it in more detail in the next video.